Hello, this is a video on the topic of silent hypoxemia as presented in some COVID-19 patients. Please read the disclaimer. You may like to stop the video while reading the disclaimer, and then you may wish to continue to watch the video. Please note, I am not a medically qualified doctor. No advice is being given, and it is not intended to give any advice. This video is for educational purposes only. This presentation is of literature search and searches on other sources such as internet and my interpretation thereof. These ideas are limited as these have not been peer reviewed. My name is Harsh Sharma. I have a BSc, MSc, PGCE, and I did a second BSc. Aims and objectives: to discuss some aspects of regulation of oxygen delivery by the blood and their possible relation to silent hypoxemia as reported for COVID-19. Target audience: university undergraduates and anyone interested in the subject with relevant prior knowledge. Oxygen delivery to the body. Some ideas towards understanding silent hypoxemia as reported for some COVID-19 patients. Hypoxemia is defined as a condition where arterial oxygen tension is below normal. Hypoxia is defined as the failure of oxygenation at the tissue level. Hypoxemia is usually defined in terms of reduced partial pressure of oxygen in arterial blood, but also in terms of reduced content of oxygen or percentage saturation of hemoglobin with oxygen. From a peer-reviewed journal, patients with coronavirus disease COVID-19 are described as exhibiting oxygen levels incompatible with life without dyspnea. More precisely termed silent hypoxemia is especially bewildering to physicians and is considered as defying basic biology. Hypoxemia produces dyspnea through the stimulation of carotid bodies which send signals to the medulla oblongata. The resulting increase in respiratory center output is transmitted down to the phrenic nerves and diaphragm, causing increased ventilation. The heightened medullary center activity is concurrently transmitted up to the cerebral cortex. It is this cortical projection that produces the unpleasant sensation of dyspnea. The chemical drive to breathe in response to hypercapnia and hypoxia exhibits as much as 300 to 600% variation between one subject and the next. This wide variability in respiratory drive is another factor that explains why some patients with hypoxia do not develop dyspnea. So we are going to discuss various factors and trying to include these aspects into our discussion. Normal arterial blood oxygenation saturation levels in humans are 95 to 100%. Some terminology I shall be using. Oxygen saturation refers to oxygen bound to hemoglobin. PaO2 refers to oxygen dissolved in arterial blood, so the A refers to arterial. PO2 is the partial pressure of oxygen dissolved in blood. PCO2 is partial pressure of carbon dioxide dissolved in blood. When we're dealing with gases dissolved in blood or oxygen bound to hemoglobin, we should be very clear which part of the body we are discussing. Lungs, arteries, veins, tissues, brain, and so on. Regulation of respiration in the chemoreceptors. This is an overall view of the regulation of respiration, and I shall be zooming in the role of chemoreceptors in regulation of respiration. Peripheral chemoreceptors are found in the carotid body and the aortic arch. Central chemoreceptors are found in the medulla oblongata in the brain. Peripheral chemoreceptors, carotid bodies, and aortic bodies. So here are the carotid bodies and the aortic bodies. And the thing to note is peripheral chemoreceptors are the only sensors detecting a fall in PO2. The carotid and aortic bodies are both sensitive to a fall in PaO2 and an increase in Pa carbon dioxide. The fall in Pa2 is detected by the carotid body and aortic body. The thing to note is they detect changes in dissolved oxygen, but not oxygen that is bound to hemoglobin. The carotid body, in addition to detecting PaO2, fall in PaO2 or increase in PaCO2, the carotid body also detects increase in hydrogen ions. So they both detect changes in oxygen or carbon dioxide in the arterial blood, and carotid body only also detects increase in hydrogen ions. This is a diagram showing this role of carotid body and aortic body in detecting changes in the blood. So they both detect a change in oxygen level, the fall in oxygen level in the arterial blood, or an increase in carbon dioxide in arterial blood, and detecting these changes, nervous impulses are generated and sent to the respiratory centers in the brain. In addition, carotid body also detects an increase in hydrogen ions, that is a decrease in pH. Notice the increase in hydrogen ions or the decrease in pH detected by carotid body can be due to a variety of reasons. For example, due to increased carbon dioxide production or increased lactic acid production. Central chemoreceptor pathways. The central chemoreceptors are an anatomical collection of neuronal chemoreceptors located just beneath the ventral surface of the brainstem's medulla, a few hundred microns away from the brainstem respiratory centers. So, the central chemoreceptors are near the respiratory centers but are not part of the respiratory centers. In the central chemoreceptors detect an increase in carbon dioxide and increase in hydrogen ions. The thing to note is the increase in carbon dioxide is detected indirectly via hydrogen ions. Under normal conditions, 75% to 85% of respiratory drive is due to stimulation of central chemoreceptors by PaCO2. The direct stimulant for neurons of central chemoreceptors is only hydrogen ions. Hydrogen ions cannot cross blood-brain barrier. Carbon dioxide can cross blood-brain barrier. So we need to discuss how central chemoreceptors are stimulated by an increase in arterial PaCO2. So this is illustrated here. This is your capillary, the blood side. This is the brain side. Carbon dioxide in the blood can diffuse into the brain. Hydrogen ions from the blood cannot enter the brain. And as we discussed, chemoreceptors, central chemoreceptors in brain can only recognize hydrogen ions and not carbon dioxide. When carbon dioxide enters brain, the enzyme carbonic anhydrase comes into play. Carbon dioxide plus water is converted to carbonic acid by the action of carbonic anhydrase. The carbonic acid then dissociates to form bicarbonate and hydrogen ion. Hydrogen ions are then created from carbon dioxide plus water by the action of carbonic anhydrase, which converts CO2 plus H2O into carbonic acid, which then dissociates to form bicarbonate ion plus hydrogen ion. Thing to notice that central chemoreceptors can detect changes in blood CO2 via the formation of hydrogen ions as described above, but the central chemoreceptors are not affected by change in blood pH, for example due to lactic acid. Another summary slide illustrating what we have discussed. This is the blood side, this is the brain side. Carbon dioxide diffuses from the blood into the brain. Hydrogen ions are generated, which are then detected by the central chemoreceptors. It appears that central chemoreceptors account for 65 to 80% of the integrated response to the respiratory acidosis under normoxic conditions. Central chemoreceptors desensitize after continuous stimulus. Respiratory drive or respiratory control refers to the process by which detected changes in the body's pH and PaCO2 levels are responded to by the central nervous system's corrective stimulation of the rhythm, effort, and rate of breathing. So this is the respiratory drive as we just discussed in the last slide. And then there is the hypoxic drive, which is the chemoreceptors in the aortic arch and carotid bodies are stimulated by a decrease in arterial oxygen concentration. Summary slide on what we have discussed. Central chemoreceptors are located on the ventrolateral surface of medulla oblongata and detect changes in the pH of spinal fluid. Peripheral chemoreceptors include the aortic body and the carotid body. Aortic body detects changes in blood oxygen and carbon dioxide, but not pH. The carotid body detects all three: blood oxygen, carbon dioxide, and pH. A cartoon illustrating what we have discussed on regulation of oxygen delivery to the body. Change in hydrogen ions is detected by carotid chemoreceptors. A change in blood CO2 levels and change in blood oxygen levels is detected by both carotid and aortic chemoreceptors. The medullary chemoreceptors, central chemoreceptors, they detect change in carbon dioxide indirectly via production of hydrogen ions. And the nerve impulses are then generated and sent to the respiratory centers in the medulla oblongata and the pons. So once a change is detected, for example an increase in arterial PCO2 by the central chemoreceptors and peripheral chemoreceptors, which then generate afferent impulses leading to increased ventilation so that more carbon dioxide is exhaled and the arterial PCO2 and pH return to normal. Decrease in arterial oxygen is detected by the peripheral chemoreceptors in carotid body and aortic arch. Again, impulses are generated, nervous impulses are generated, leading to events to change in alveolar ventilation rate, returning the PO2 back to normal. Carbon dioxide diffuses much more readily than oxygen. So it is possible that many aspects of lung pathology would have a lesser effect on carbon dioxide diffusion out of lungs and more effect on oxygen diffusion into lungs. Let's look at the oxygen and carbon dioxide. Normally, the arterial PO2 equals 100 mm of mercury and arterial CO2 equals 40 mm of mercury. So the arterial PO2 is 100 mm of mercury and the arterial PCO2 is 40 mm of mercury. We appreciate that we need the right amount of oxygen in the blood. We also need to appreciate that we need the right amount of carbon dioxide in the blood also. Too little carbon dioxide leads to hypocapnia, too much carbon dioxide leads to hypercapnia, and both hypocapnia and hypercapnia create problems and pathological changes for the patient. This represents the right amount of carbon dioxide in the blood, and if there is increase in carbon dioxide levels in the blood, one of the outcomes is increased cerebral blood flow, which will cause problems. Or if there is a decrease in carbon dioxide level in the blood, one of the outcomes is decreased cerebral blood flow. Too much carbon dioxide in the blood can lead to respiratory acidosis. Too little carbon dioxide in the blood can lead to respiratory alkalosis. This is a graph showing the effect of arterial PCO2 on the cerebral blood flow. The normal arterial PCO2 is around 40 mm of mercury. At arterial PCO2 of 40 mm of mercury, the normal blood flow is 50 ml per minute for 100 gram of tissue. That is 50 ml per minute for 100 gram of brain tissue. When we go above 40 mm of mercury, PCO2, there is hypocapnia, and when there is PCO2 going below 40 mm of mercury, we are getting hypocapnia. This is a cartoon showing consequences of hypocapnia. There is decrease in cerebral blood flow. Vasoconstriction brought upon by hypocapnia can cause a reduction in cerebral blood flow, resulting in ischemic hypoxia. There is decreased cerebral blood volume and decrease in intracranial pressure. decrease in intracranial pressure, there is decreased oxygenation. Hypocapnia can cause a shift in the percent saturation hemoglobin curve to the left. The oxygen hemoglobin dissociation curve can shift to the left as a result of hypocapnia, which increases the hemoglobin's affinity for oxygen and hinders the release of oxygen into the cells, which is known as high affinity hypoxia. We will see this in more detail later on. Increased metabolism. Heightened neuronal excitability and cerebral metabolism brought upon by hypocapnia increases metabolic needs, resulting in hypoxia. So, with a detection of increasing of arterial PCO2 by the central chemoreceptors or the peripheral chemoreceptors, afferent impulses will be generated, leading to events of increased ventilation so that more carbon dioxide is exhaled and returning the arterial PCO2 and pH back to normal. With a decrease in PO2, this is detected by the peripheral chemoreceptors in carotid and aortic arch. Again, nervous impulses will be generated that will lead to change in alveolar ventilation, returning the oxygen levels back to normal in the blood. The thing to note is that increased ventilation can get rid of CO2 to such an extent to cause hypocapnia. So, when there is a need to increase ventilation, and ventilation as required will increase, but it can then go on to get rid of too much carbon dioxide to such an extent to cause hypocapnia. Hypocapnia reduces cerebral blood flow and reduces blood flow to many other organs. So it's not just the cerebral blood flow that is affected, blood flow to other organs is also reduced due to hypocapnia. A graph showing the effect of change in arterial PO2 on cerebral blood flow. The normal arterial PO2 is 100 mm of mercury. As we decrease the arterial PO2 to until about 60 mm of mercury, then after 60 mm of mercury there is a sharp rise in cerebral blood flow. So not much happens with regards to cerebral blood flow as we drop from 100 mm of mercury to 60 mm of mercury. Other things may be happening, indeed other things are happening, but the cerebral blood flow is not affected until the arterial PO2 decreases below 60 mm of mercury. Compare this with carbon dioxide. As the arterial PCO2 decreases, straightway there is a decline in cerebral blood flow. Whereas with oxygen, as the amount of oxygen decreases in blood, first of all there is no effect until a decrease or up to 60 mm of mercury in the arterial blood, and then there is a sharp increase in the cerebral blood flow. On this slide we see the effect of drop in arterial PO2 on the alveolar ventilation. As the arterial PO2 decreases from 100 downwards, straightway there is an increase in alveolar ventilation, and this increase becomes more marked once the arterial PO2 has dropped below 60. So there is a more sharper increase in alveolar ventilation after the arterial PO2 drops below 60, but Even then, soon as it starts to drop below 100, the arterial PO2, there is an increase of some kind in the alveolar ventilation. And a reminder, a fall in PO2 is detected by the peripheral chemoreceptors in the carotid body and the aortic body. A summary slide showing what we have discussed on the effect of change in arterial PO2 and PCO2 on cerebral blood flow. The normal arterial PO2 is 100 mm of mercury. When that comes down to about 60 mm of mercury, there is effectively no change in the cerebral blood flow. Once we drop below 60 mm of mercury, that is, once the arterial PO2 drops below 60 mm of mercury, then there is a sharp increase in cerebral blood flow. Compare that to the arterial PCO2, which is normal. The normal PCO2, arterial PCO2, is 40 mm of mercury. Soon as this comes down, soon as you go below the normal arterial PCO2 of 40 mm of mercury, there is a gradual decrease in cerebral blood flow. This is a graph of change in arterial PO2 and the alveolar ventilation. As we decrease the arterial PO2 from normal of 100 mm of mercury, there is an increase in alveolar ventilation. But when we reach about 60, this increase is more sharp. So first there is a gradual increase in alveolar ventilation, and that becomes more significant once the arterial PO2 has dropped to 60 mm of mercury. A reminder, peripheral chemoreceptors are the only sensors detecting a fall in PO2. Hemoglobin and oxygen delivery to the tissues. This is a hemoglobin saturation curve, that is percent O2 saturation of hemoglobin. On this axis is the PO2 in the blood, and in the lungs we will see there's a PO2 of 100, that is in the normal lungs. In the tissues such as muscles and so on, where oxygen is being used, the PO2 falls down to about 40 mm of mercury. And you can see that percent O2 saturation, so how much oxygen is bound to hemoglobin, depends on the PO2 in the blood. So at the PO2 of 100 mm of mercury, which is found in the blood in the lungs, hemoglobin is 100% saturated. In the tissues, where the PO2 has fallen down to 40, the hemoglobin is now less saturated, about 75% saturation. So it has now given up its oxygen. There it was 100% saturated, now it is only 75% saturated here. So the hemoglobin has given up its oxygen to the tissues, so it is now less saturated than before. So in the lungs, hemoglobin is 100% saturated. It picks up oxygen in the lungs. Then the blood comes to the tissues where PO2 is lower, and at this lower PO2 of blood, hemoglobin gives off its oxygen. So the hemoglobin oxygen binding curve can be affected by many factors. This slide shows the effect of hypocapnia and hypercapnia. Hypocapnia, that is too little carbon dioxide in the blood, when there is a decrease in carbon dioxide in the blood, the hemoglobin oxygen binding curve is shifted to the left. If there is too much carbon dioxide in the blood, hemoglobin oxygen binding curve is shifted to the right. So there are other factors here that can affect the shifting of the hemoglobin oxygen binding curve. These factors are again given in a table form. So let's have a look at what happens when the curve is shifted to the left or to the right. Let's have a look at some numbers. So we are going to look at saturation of hemoglobin in the tissues when PO2 is 30 millimeter of mercury. So here it is, 30 millimeter of mercury, PO2. This is what you will find in the tissues, and we are going to see what happens in the case of hypocapnia, normal physiological conditions, and hypercapnia. But first, take a look at the hemoglobin binding to oxygen when PO2 is 100 millimeter of mercury. This is the situation in lung, and in particular take note, we are talking about the normal lung. This is the normal lung, PO2 of 100 millimeter of mercury, and under the three situations, there's very little effect on the binding of oxygen to hemoglobin, whether there is a hypocapnia, hypercapnia, or normal physiological conditions. So the way to look at this is, if you look at the vertical distance between the three curves, there is very little difference. So these three curves are very close together when you look at the vertical distance between them. But what happens when the PO2 is 30 millimeter of mercury, as you will find in tissues? There, when we compare the vertical distance between the curves, it is quite significant. With hypercapnia, hemoglobin is 78% saturated. Under normal physiological conditions, hemoglobin is 63% saturated. With hypercapnia, hemoglobin is 44% saturated. So the saturation of hemoglobin at 30 millimeter of mercury, at PO2 of 30 millimeter of mercury, changes whether there is hypercapnia or normal physiological conditions or hypercapnia. Now the important thing to note is here the hemoglobin is more saturated than normal and certainly than hypercapnia. So hypercapnia hinders release of oxygen from the hemoglobin to the tissues. That is why it remains more highly saturated. Hypercapnia, in fact, helps unloading of oxygen from hemoglobin, and that is why it is less saturated. So in the tissues where the PO2 is 30 millimeter of mercury, hypercapnia causes more release of oxygen from hemoglobin, so it is less saturated than normal physiological, and hypercapnia hinders release of oxygen from the hemoglobin, so it remains more saturated as compared to normal or hypercapnia situation. More saturated hemoglobin is in the tissues, less oxygen is given to the tissues, and more is taken back to the lungs. So it is less oxygen that is given to the tissues. Increased affinity of hemoglobin for oxygen may increase blood saturation of hemoglobin. However, we need to consider usefulness of this in terms of delivery of oxygen to the tissues. So now let's have a look, try to bring this together in the case of silent hypoxemia. This is a screenshot taken from video by Shiv Kumar Singh, and there it is suggested that in COVID-19 patients, carbon dioxide levels does not fall too much and is maintained at near normal. So hypocapnia is avoided. So there we are. Examples are given where the PCO2 is nearly normal at about 40 millimeter of mercury, maybe slightly less, maybe slightly more, but it's nearly normal. I think we can accept that. And the cerebral blood flow is again nearly normal. Not quite, but nearly normal. It is suggested the lack of oxygen should be detected by peripheral chemoreceptors and ventilation increased. This does not happen, possibly due to nerve impairment caused by SARS-CoV-2. Although hypoxia remains, hypocapnia does not ensue. Thus, there is no cerebral vasoconstriction. So this is an idea which is worth considering, that in these COVID patients that are showing silent hypoxemia, there is not hypocapnia that affects the cerebral blood flow, and the lack of oxygen is not detected by the peripheral chemoreceptors, maybe due to some kind of nerve damage. So it is suggested that the patients do not develop hypocapnia and ensuing pathology. However, situation for the patient is very serious owing to low oxygen blood saturation. Other factors. Hemoglobin oxygen affinity can be affected by several other factors, and an important one is 2,3-bisphosphoglycerate, 2,3-BPG, which is the same as 2,3-diphosphoglycerate, 2,3-DPG. I shall be using these terms interchangeably. The thing to remember is 2,3-DPG is the older terminology, 2,3-BPG is the current correct terminology. In the red blood cells, there is a normal concentration of 2,3-diphosphoglycerate. Okay, so there is present in the red blood cells 2,3-diphosphoglycerate at all times. Hypoxic conditions, for example, high altitude, will increase 2,3-diphosphoglycerate in red blood cells. So this is shown on the graph. Normal hemoglobin oxygen binding curve at the normal blood 2,3-bisphosphoglycerate, which is 5 millimole per liter. So at normal concentration of 2,3-BPG, this is the oxygen hemoglobin binding curve, percent saturation of hemoglobin with oxygen. When 2,3-BPG is increased, for example, when somebody goes to high altitude, the curve is shifted to the right. This is an experimental condition where in the test tube, if you remove all 2,3-bisphosphoglycerate from hemoglobin, you can see the oxygen hemoglobin affinity is hemoglobin has much higher affinity for oxygen. And the curve is no longer sigmoid. This is a sigmoid shaped curve. When 2,3-BPG is completely removed, the hemoglobin then no longer displays a sigmoid curve, as you can see here. For completeness, I have included myoglobin oxygen binding curve in comparison to the hemoglobin oxygen binding curve. Myoglobin has a very high affinity for oxygen even at very low partial pressures. This means oxymyoglobin will only dissociate when oxygen levels are low. It is found in muscle cells where it acts as an oxygen reserve. Myoglobin is a heme-containing globular protein that is found in abundance in myocyte cells of heart and skeletal muscle. So, look at the PO2 in millimeter of mercury as in muscles which are doing strenuous exercise. So this is the PO2 probably going to be in a muscle which is doing strenuous exercise. Notice even when the PO2 is less than 10 millimeter of mercury, myoglobin is still holding on to most of its oxygen. It is when the PO2 drops very low, myoglobin then gives up its oxygen. So remember, less saturated the myoglobin is, more oxygen it has given off. More saturated the myoglobin is, more fraction of sites that are bound, the more myoglobin is holding on to its oxygen and it's not giving it up. So up here, less fraction sites are bound, so myoglobin is now giving up. But remember, it is at very low PO2. Otherwise, it holds on to its oxygen. The thing to note is this is under physiological conditions. So this is under physiological conditions. Just a reminder that the myoglobin oxygen binding curve here is under physiological conditions. And look at this black curve, which is hemoglobin stripped of 2,3-bisphosphoglycerate under experimental conditions. So this black curve of hemoglobin is under experimental conditions, not physiological conditions. And the blue curve is normal physiological conditions, and red curve is physiological conditions under which is more hypoxic. Okay, so the red curve is when there is some kind of hypoxia, a need of oxygen there, that 2,3-bisphosphoglycerate increases in the red blood cells, which causes a shift of the hemoglobin oxygen binding curve to the right. But this is in the body, whereas this black curve of hemoglobin oxygen binding with 2,3-bisphosphoglycerate removed is in the test tube under experimental conditions. In comparison, the myoglobin oxygen binding curve is under normal physiological conditions, or which may change. That is to say, this is the situation in strenuous exercising muscle. So this is, we will be looking at this curve now, and we will look at in detail the effect of this shift of the dissociation curve due to increase in 2,3-diphosphoglycerate. Again, this is the PO2 millimeter of mercury, and this is the hemoglobin saturation, percentage saturation of hemoglobin. In essence, when the curve is shifted to the right due to extra 2,3-DPG, there is very little effect on the ability of hemoglobin to pick up oxygen in the lungs, and emphasizing we are talking about normal lungs. Unloading of oxygen in the tissues is much more efficient in presence of extra 2,3-diphosphoglycerate. So when the curve is shifted to the right, in the tissues where there is low PO2, oxygen is given off much more easily by hemoglobin. So let's have a look at partial pressure of oxygen of 38 millimeter of mercury and compare to, for example, 100 millimeter of mercury to see there's very little difference in the two curves. The vertical drop between the two curves is very little. So in this area, in the area of normal lung, both curves are similar in their ability to pick up oxygen. So hemoglobin is affected very little by the extra 2,3-DPG in its ability to pick up oxygen at a higher PO2 around the 100 millimeter of mercury that will be found in normal lungs. But when we look at PO2 of 38 millimeter of mercury, and with normal levels of 2,3 dpg, hemoglobin is around 70% saturated. So at normal physiological conditions with the normal level of 2,3 dpg, when the PO2 is 38 millimeter of mercury, as will be found in the tissues, hemoglobin is 70% saturated. But when there is extra 2,3 dpg, then at the same PO2, 38 millimeter of mercury, hemoglobin is now 45% saturated. So with extra 2,3 dpg present, hemoglobin is now only 45% saturated. The reason it is less saturated now is that more oxygen has been delivered to the tissues. So up here with the normal 2,3 dpg, hemoglobin is still able to hold on to a fair amount of oxygen. But when the 2,3 dpg is increased, hemoglobin is only 45% saturated, less saturated. That means more oxygen has been delivered to the tissues. So in presence of increased 2,3 diphosphoglycerate, hemoglobin is 45% saturated compared 70% in normal times. Less saturated hemoglobin means more oxygen has been delivered to the tissues. So again, just a summary slide reminding us very little effect on ability of hemoglobin to pick up oxygen in presence of extra 2,3 diphosphoglycerate. Unloading of oxygen in tissues is much more efficient in presence of extra 2,3 diphosphoglycerate. Although more oxygen has been delivered to the tissues, now there is less oxygen bound to hemoglobin. It's a question I'm asking. I do not know the answer to that, but it's worth asking the question, does this mean there is reduced reserve capacity? There is less oxygen left over bound to hemoglobin to meet any possible desperate needs. Just to mention, the increase in 2,3 diphosphoglycerate takes place within a day or even hours. Hemoglobin binding to oxygen is affected by many other parameters not discussed here. For example, pH, temperature. In many cases, COVID-19 patients' temperature is increased. Ventilation and perfusion. When the alveoli are well ventilated, they are well perfused. If the alveolus is not properly ventilated, then the blood flow to it is reduced due to vasoconstriction, so that more blood can be directed to a well ventilated alveolus. So capillaries are constricted in poorly ventilated areas to divert blood to areas of well ventilated area. There can be sometimes mismatching of blood flow and alveolar ventilation. In acute respiratory distress syndrome, alveolar edema may be caused by capillary leaking and dilation. Hence, more blood, dilated capillary, is sent to poorly ventilated regions of the lung. Less blood goes to the well ventilated area. So we have more blood going to the poorly ventilated area compared to a well ventilated area. Now we will look at other effects, such as one of them is increase in red blood cell production. Hypoxic conditions are sensed by the liver and the kidneys, which will release erythropoitin that will lead to increased red blood cell production. Increase in red blood cells will help deliver oxygen to the tissues, but there are concerns. Increased red blood cells may increase blood viscosity and cause congestion in the lungs where there is already existing pathology, and can cause congestion in other tissues, especially kidneys. Erythropoitin-induced increase in red blood cells takes days, weeks to take full effect. So let's have a look at the issues in the kidney. So there is your blood coming to the glomerulus in the kidney, and 20% of the liquid will be filtered. So the blood going to the afferent arteriole is now bathed in 80% of the plasma volume. So the same number of red blood cells are now contained within a lesser volume of fluid. So this obviously increases the viscosity of the blood. The fluid will be reabsorbed, bringing the blood viscosity back to normal. But in this portion of the glomerulus, in this portion of the kidney, the afferent arteriole, the blood viscosity is higher because the fluid has been filtered. So if the blood is already increased in viscosity due to increased number of red blood cells, then the fluid is filtered in the glomerulus, and now that there is a further increase in blood viscosity due to glomerular filtration. So you have a more viscous blood due to increased number of red blood cells already, and then further increase in viscosity due to glomerular filtration. So if the blood is already increased in viscosity due to increased red cell production, this can cause congestion in the afferent arteriole. Under normal circumstances, blood in afferent arteriole is much more viscous than normal. It will normalize by reabsorption of water in peritubular capillaries. And recapping that if the blood is already increased in viscosity due to increased red cell production, this can cause congestion in the afferent arteriole. Systemic vascular response to hypoxia. There is a systemic vasodilation. Not all capillaries have blood flowing through when at rest or low demand by organs for oxygen. Hence, there is a reserve of capillaries that open up when there is demand. What could be the cost? This will cause drop in blood pressure. Many COVID-19 patients are already in sepsis with hypovolemia. So now are the summary diagrams, oxygen regulation and flip side. So CO2 diffusion is easier than oxygen, that maintains pH balance. Increase within red blood cells of 2,3-diphosphoglycerate, an altering of hemoglobin oxygen binding curve, dissociation curve, better delivery of oxygen to tissues. Increase red cell production, better delivery of oxygen. Oxygen regulation, flip side. CO2 diffusion from blood into alveolus may be less affected than oxygen diffusion from lung into the capillary. The thing to remember is that 75 to 85% of the respiratory drive is due to carbon dioxide stimulating the central chemoreceptors, that is carbon dioxide via generation of hydrogen ions. So if we do not get a buildup of carbon dioxide but oxygen is getting low, the regulatory system may not detect this, so there may be a false sense of security, unaware that oxygen supply is short. On the other hand, there is a possibility of hypocapnia and associated pathology. Increase within red blood cells of 2,3-diphosphoglycerate, an altering of hemoglobin, oxygen, dissociation curve. A lot more oxygen has been unloaded, and there may be a situation that hemoglobin has reduced reserve capacity to deliver oxygen. increased red cell production. This adds to congestion in lungs, in ARDS, and elsewhere, for example, kidney. So this is putting it all together, the oxygen regulation and the flip side. So there is a fine balance seesaw. You have factors which are helping to deliver oxygen to the body under these conditions, but then there's a cost of these factors. For example, erythropoitin will increase red blood cell synthesis, which will help to deliver oxygen, but the other side is erythropoitin increased red blood cell synthesis can lead to increased viscosity of the blood. Similarly, opening up of capillaries to deliver more blood can lead to reduced blood pressure. Increase in 2,3-diphosphoglycerate, more unloading of oxygen in the tissues, there is less reserve capacity of oxygen left bound to hemoglobin. No hypocapnia, no cerebral blood flow restriction, no increase in hemoglobin affinity for oxygen, but there is ongoing hypoxia. Add to this damage and recovery to the organs, especially lungs. The seesaw is very finely balanced. We hope the balance will be in favor of patient recovery. We have discussed hemoglobin oxygen saturation in normal lung. When this oxygen partial pressure is 100 mm mercury as it is in normal lung, the binding of hemoglobin to oxygen is hardly affected. There is very little effect whether there is hypocapnia or there is hypercapnia. That is in the normal lung. I do not know the answer to this question, but it needs to be answered. What is a PO2 in a COVID-19 patient with acute respiratory distress syndrome, for example, in that patient's lung? Is also worth discussing that PO2 in a COVID-19 ARDS lung may vary in different parts of the lung. For example, there may be edema here leading to low PO2. This may be relatively normal or relatively less affected, and PO2 may be nearly normal in this portion of the lung. So we need full discussion on these aspects and the consequences of this different partial pressures of oxygen in different parts of the lung, if that is the case. We have discussed several parameters that can affect hemoglobin oxygen saturation. We discussed hypocapnia, decrease in PCO2, increase in 2,3-diphosphoglycerate. Can shift it to the right. Hypocapnia, decrease in PCO2 will shift the curve to the left. But in a patient, several factors may be taking place concurrently, simultaneously. These are some of the few factors shown. This is not a comprehensive slide. So we need to discuss all the factors that could be taking place, some acting in one direction, some acting in the other direction. What could be the overall effect? We need test tube experimental data to find all this out. Thank you.